Hello students, this is Dr. Ravi Sani. Today we are going to discuss about the mechanism of muscle contraction. So basically we are going to discuss about the specific changes which occurs or which are responsible for contraction of muscle. That is really important concept and which usually asked in various exams. Now come to the mechanism of muscle contraction. So basically, according to sliding filament theory, contraction of muscle fibers occurs by the sliding of thin filaments over the thick filament. So basically, there is a sliding filament theory according to which a contraction of muscle fibers occurs by sliding of thin filament. We studied in previous lectures that actin filament is responsible for the formation of thin filament, actin protein, and in case of thick filament, which is made up by the myosin protein. So there is a sliding of that actin filament over the myosin filament. In this image, we can easily appreciate this is the thin filament, which is made up of various proteins like troponin, actin, tropomyosin, and in case of myosin protein, which made up a thick filament, there is a tail portion and head portion. This head portion got attached with the actin protein. This myosin head basically attached to the actin uh, protein, where there is a actin binding site and ATP binding site, because sliding of actin filament over the myosin filament requires ATP. So various steps. An impulse from CNS reaches the neuromuscular junction, basically at the motor end plate via motor neuron. We have studied about the neuromuscular junction, that it is a synapse between motor neuron and a sarcolemma of muscle fiber. So neuromuscular junction is a junction between or nerve cells and the muscle fibers. And at microscopic level, we can call it is a synapse between motor neuron and sarcolemma of muscle fiber. So basically, a motor neuron plus muscle fiber, we can call it a motor unit. This is a neuromuscular junction, nerve cells, muscle fibers, muscle cell, and this specific portion we are calling it neuromuscular junction. These are the various microscopic changes which we will study in next slides. So whenever that nerve impulse travels to the sarcolemma, by this, when a nerve impulse travels from this nerve cells to the sarcolemma of muscle cells, these are the synaptic vesicles at the terminal end. So synaptic vesicles basically releases neurotransmitters. We are calling it acetylcholine. We can understand in a very simple language that basically action potential, is traveling from nerve cells, and this action potential, basically, what is action potential? Action potential, it is changes, electrical changes across the membrane. In normal state, cell is in polarized condition. So any electrical changes which reverse this polarity could be responsible for the generation of action potential. This action potential travels from nerves or neuron, and when it reaches to this terminal end, there are synaptic vesicles. Vesicles. These synaptic vesicles releases acetylcholine, and this acetylcholine is responsible for the opening of sodium channels. These sodium channels will generate action potential in sarcolemma, and this action potential, which is generated in the sarcolemma, spreads rapidly through the muscle fiber, and this will finally cause release of calcium ions from sarcotubular or the sarcoplasmic cisternae. There is a sarcotubular system in which we have tried. There are L tubules and T tubules. So basically, one T tubules and two L tubules forms a triad portion, forms a triad, which is responsible for the formation of sarcotubular system. And in this, we have calcium channels, which will be responsible for the release of calcium from these channels. And this calcium binds with troponin on actin filament. This is the actin filament. There is a troponin protein. This troponin protein or subunit on actin filament got unmasked by the binding of calcium. When action potential generated in the uh, nerve cell or nerves, it got traveled to the terminal portion where it was responsible for the generation, it was responsible for the release of acetylcholine from these vesicles. Acetylcholine got released. This neurotransmitter got attached with the sarcolemma of the muscle, and which was responsible for the opening of sodium channels. And this sodium channel again will generate action potential in muscle cell or the sarcolemma of the muscle fiber, and this will release calcium ions. This calcium will bind with the troponin subunit and unmask the actin site of myosin. Sorry, that is active site of the myosin. There were also ATP binding site also, which was responsible for the which was responsible for the binding of myosin filament. That myosin head binds to active sites and forms cross bridges by this ATP hydrolysis. This generation of ATP will results in movement of this myosin head. Basically, there is a pull of the actin filament on both sides of the A band, and actin filaments partially overlaps, and finally H zone disappears. The Z lines attached to actins is pull inward. When calcium binded with the this tropomyosin protein, sorry, that troponin protein, and unmasked the active site, which was responsible for the binding of myosin to the actin filament, and ATP hydrolysis finally resulted into the movement of the myosin filament over the actin filament. And due to this contraction, basically this Z line got when contraction occurs, this H zone got shrunken. The width of this S zone decreased because on the both the directions myosin filaments are stretching or they are pulling actin filaments inwards. So it causes shortening of sarcomere. The Z lines attached to the actins also pull inward. This Z line also got pulled inward. H zone, the width of H zone is also decreased because these are the myosin filaments which pulled actin filament inward by the help of ATP hydrolysis, and this was occurred due to release of calcium by the sarcotubular system which was present at the muscle fibers, and this was due to the generation of action potential by the action potential from nerve cells, which was responsible for the release of acetylcholine. This acetylcholine was responsible for the generation of action potential on the muscle fiber or muscle cell. Myosin releases the ATP and phosphate and goes back to its relaxed state. Now, a new ATP binds again, and the cross bridge is broken. So this cycle repeated again and again. When calcium ions are pumped back to the sarcoplasmic cisternae, the actin filaments are again masked. As a result, the Z line returns to their original position. We are calling it relaxation. When calcium ions pumped back in the sarcotubular system, the actin filaments again got masked by the troponin protein, and the Z lines returned to their original positions. This zone got the width of this zone reduced. Again, it is gaining its uh, natural position. The width of these bands also got at the previous or the normal position. The reaction time of the fibers varies in different muscles. Repeated activation of muscle leads to the accumulation of lactic acids. This is responsible for the muscle fatigue. The production of lactic acid or the accumulation of lactic acid is responsible for the muscle fatigue. Basically, it is due to the anaerobic breakdown of the glycogen and aerobic breakdown of the glycogen. During exercise, basically, there are two components, the aerobic and the anaerobic component. And the anaerobic component is responsible for the breakdown of the glycogen because aerobic portion is quite small. These are the differences between red muscle fibers and white muscle fibers. Red colored due to myoglobin, more mitochondria are present, aerobic metabolism is present, slow and sustained contractions. The opposite conditions are present in white muscle fibers, lesser myoglobin, less mitochondria, and aerobic